Okay, good morning, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. It is a great pleasure to welcome you to this webinar on Applied Microbiology Series 32 with topic Application of Cell-Free Protein Synthesis Systems for Antibody, Enzyme, and Peptide Engineering, held by Research Center for Applied Microbiology, National Research and Innovation Agency BRIN, today, Wednesday, June 12, 2024. You can also watch this webinar on YouTube at PR Mikrobiologi Terapan BRIN channel. In this occasion, we are honored to welcome Dr. Ahmad Fatoni M.Eng, Head of Research Center for Applied Microbiology BRIN. Our speaker, Professor Hideo Nakano, Professor at Graduate School of Bioagricultural Sciences, Nagoya University, Japan. And our moderator for today, Dr. Renat Linda Sukmarini M.Eng, researcher from Research Center for Applied Microbiology BRIN. Ladies and gentlemen, next agenda is singing the national anthem, Indonesia Raya. Panitia bisa di-share Indonesia Raya?
Siapanya?
Mas Rian? Oke. Ladies and gentlemen, next is opening remark from Head of Research Center for Applied Microbiology BRIN. To Dr. Ahmad Fatoni, time is yours.
Okay, thank you, Miss Uci. Tes. Oke ya, oke. My voice clear? Izin? Oke ya, oke. Okay, thank you, everyone. Honorable Speaker, Professor Hideo Nakano of Graduate School of Agricultural Science, University of Nagoya, Japan, and his team, and then Bapak Ibu sekalian, everyone, researchers from BRIN or university. Assalamualaikum warahmatullahi wabarakatuh. Very good morning, everyone. Thank you very much for your attendance and First of all, I would like to welcome Professor Hideo Nakano because this is his first time visit to BRIN. So he's now with us here at Soekarno Science and Technology Park in Cipinong. And after this seminar, we hope that we can discuss further about potential future collaboration between Nagoya University and BRIN, or National Research and Innovation Agency. Yeah, thank you very much, Prof. Nakano, for accepting our invitation and then later on discuss about our future collaboration. And today we will have a topic of application of cell-free protein synthesis systems for antibody, enzyme, and peptide engineering. I think this topic is very linear with our focus in BRIN. We have 12 research organizations and about 85 research centers, and also we have several research centers who are working on uh, uh, protein for antibody, enzyme, and peptide. And here with us, Prof. Nakano, we have several research group that we invited, focus on enzyme and peptide. Hopefully, after this meeting, we can discuss further, and then we will also have lab visit around Cipinong, and then hopefully from the visit today and the discussion today, we will develop the idea what kind of research that we can collaborate with between BRIN and Nagoya University. And as also for the information for Prof. Nakano and team, because this is the first time for them to visit BRIN. BRIN actually has several schemes, Prof. Nakano, yeah, that can be utilized to support our future collaboration. So like, for example, for research facilitation, we have several schemes that we can propose together, we can make a joint proposal and then apply it to BRIN to run our research collaboration. And also we have mobility program. So in this case, we can do the joint supervision for PhD student, for example, and then also we can invite professor and team from Nagoya to BRIN here, working together with us for like 1 or 3 months, for example. And also we also have joint research visits. So we can send actually our staff, our researcher to Nagoya University, to your laboratory, working together with your team there, and then do the research together. And currently, Prof. Nakano, maybe this is quite interesting as well, that we are now in the process of proposal selection. We have a collaboration platform, we call it structural biology on biomolecule from Indonesian biodiversity. So this afternoon you will see visit our KRIM facility in Cipinong. So to support the activity, our infrastructure is quite complete and advanced as well. And then I hope that we can soon set up the collaboration on a specific topic, specifically in structural biology, or your expertise in protein synthesis for antibody, enzyme, and peptide engineering. Yeah. And also I also invite everyone here from university as well to collaborate with us, because BRIN is not for BRIN, but BRIN for national. This is for everyone, including university and industry are very welcome to collaborate with us, and then also to utilize our facility or infrastructure. And so because here with us here with we have one lecturer from Mataram University, so I think this is a good opportunity for us. So please do collaboration. So I'm sure we can have like a strong and also long collaboration on specifically life science research here in Cipinong. Yeah, I think that's all my speech that I can deliver this morning. I hope again everyone. So please do the discussion with our speaker, Prof. Nakano. So he is the expert on the protein synthesis, and I'm sure that in BRIN we have many researchers who are working on this field. Yeah. So please use this opportunity, take this opportunity to expand your network, and then also we can soon set up the collaboration agreement, yeah, between Nagoya and Purin. Yeah, okay. And also by saying Bismillahirrahmanirrahim, I officially open this sharing session or this webinar. Thank you, and enjoy everyone for the talk by Prof. Nakano. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mr. Fatoni. Ladies and gentlemen, before we start the agenda, we will have a photo session first. To all participants, please activate your camera, and for the committee to take the screenshot. Okay, I will start count from first slide. Ready? 1, 2, 3. Second slide. 1, 2, 3. Third slide. 1, 2, 3. Thank you.
Sorry.
And I would like to remind to all participants, please fill the attendance form from the link provided by the committee in the chat feature. Now we are going to start our discussion for today, which will be led by Dr. Renat Linda Sukmarini, MN. So Mrs. Linda, time is yours.
Thank you, Mbak Suci, for bringing us to a very productive session today. Good morning, all. Honorable Head of Research Center for Applied Microbiology, Dr. Ahmad Fatoni. Thank you, Pak, for a nice opening speech. Our distinguished speaker from Nagoya University, Japan, Professor Hideo Nakano, and all audience researchers from BRIN or university, Bapak and Ibu. Assalamualaikum warahmatullahi wabarakatuh. I am Linda Sukmarini. It gives me a great pleasure to be the moderator for the webinar series on applied microbiology with the topic on cell-free protein synthesis. As we know that cell-free protein synthesis is emerging as a powerful tool to accelerate the progress of synthetic biology. In today's webinar series, we will get a closer look into application of cell-free protein synthesis systems for antibody, enzyme, and peptide engineering. Bapak and Ibu participants, first, I would like to read a brief CV of our distinguished speaker, Prof. Hideo Nakano. Yeah. He's a professor from Graduate School of Bioagricultural Science, Nagoya University, Japan, and he is also technical advisor of iBody Inc. He has personal interest in innovative biotechnology, enzyme bioprocessing, and entrepreneurial education. And he received his PhD in chemical engineering from the University of Tokyo, Japan. And he has been did some visiting research fellow from some prestigious universities such as Stanford University and UK Berkeley in the US. He also had of some and board member of some scientific society in Japan. He has also a lot of many international publication of his work. Okay. Before the presentation begins, let me kindly remind you to keep your mic on mute during the presentation. And please give your full attention to this webinar. I'm sure all of us here will be able to gain knowledge and learn more about today's topic. And last but not least, if any of you have a question, please hold on to your question because we will be having a Q&A session at the end of this webinar. Or you can just type in the chat box to post your question. And our team will be monitored by all posting in the chat box. And so please don't be hesitate to ask. Thank you. Okay. To kick off this webinar, without any further ado, I would like to welcome our today's special speaker, Professor Hideo Nakano, to deliver the presentation. All right, Professor Nakano, the screen is yours now.
Okay, thank you very much for your kind introduction. I'm very happy to be here to have a chance to visit BRIN, and also have a chance to share our research result and now past and current. Okay, I'd like to share my slides first. So okay, so here. Okay, so okay, this is my title today. So this is a photo of Nagoya Castle. Actually, I'm from Nagoya University. If you have a chance to visit Nagoya, please enjoy it. So, okay, so this is today's content. So first of all, I'd like to give a short overview about cell-free protein synthesis. And after that, I would like to talk about several topics about the application. The first, I'd like to talk about antibody screening and peptide screening and enzyme substrate screening. And in the end, I'd like to talk about the very interesting translation enhancing that we have found. So I'd like to talk it in the last. So first of all, what is cell-free protein synthesis? It is actually in vitro system that make the protein directly from DNA, messenger RNA, and protein or peptide. So normally, the E. coli cell extracts or currently purified all the components necessary for translation purified and ribosome. So in addition to that, of course, ATP, GTP, CTP, UTP should be added and amino acid and ATP regeneration system. And also depend on the protein that you want to make, it's better to add some chaperone to help the protein folding. The old process is done in the cell-free system, therefore, so you don't need to use living cells, E. coli anymore. So what is the advantage of the cell-free protein synthesis? First of all, even a PCA product can be used as a template. Therefore, so no cloning is required. The reaction to make a protein is finished within several hours. So you can get it anytime you need. In addition, control of the reaction condition is relatively easy. It's a kind of open system. You can add something that you require necessary for a protein synthesis or the protein folding. And in addition to, incorporation of unnatural amino acid is possible. So using very cutting edge technologies, you can increase the number of amino acid from 20 to much more. And the reaction minimization of the reaction scale is relatively easy, so that you can control it as you like. So actually, this is this technology is originally found, is established in 1960s. It's actually a very old technology. And then recently, there are several startups was made from many universities, especially from Japan and United States. And especially now the gene frontier is actually from Professor Takuya Ueda, the Tokyo University. This company is special because they provide a purified, reconstitute E. coli cell-free system. That is actually a laboratory use this system. And also, Peptide is actually one of the company already listed company in Tokyo stock market. And this company also established by the Professor Suga, professor in Tokyo University, and very excellent technology using cell-free system and making a specific binder to target molecules. And also, Stanford James Schwartz, actually I stay his laboratory kind of short time, and he also make a startup to produce a kind of a medicine from using cell-free protein synthesis. And also Jim Sweat, now he moved to Stanford, and the successor of Jim Schwartz, and he make the cell-free startup using cell-free system and solid already. And hybrid incorporation, actually, this is company is made by me and Dr. Kato, and main business is that a screening of monoclonal antibody using cell-free systems. So, okay, so then I would like to talk by my own research about antibody, monoclonal antibody using cell-free system. You know, monoclonal antibody is currently very widely used in many fields like research, biomonitoring, and diagnostics, and medicine. So among these, the market of the antibody medicine is so huge and still going very rapidly. As you can see, that's a total market size in 2023 is 247 billion US dollars, and it's growing very fast, so huge. So one of the reasons that so many bio medicine is use antibody, monoclonal antibody as platform modality. So one of the very famous is that Professor Honjo Tasuku got the Nobel Prize for finding monoclonal antibody to cure the cancer cell very effectively. But to make a monoclonal antibody is not easy even now. The reason is that we cannot easily get the monoclonal antibody. So traditionally, you know, the hybridoma system have been used many years. So to get the monoclonal antibody using hybridoma, first you have to immunize the antigen to mice, and then sacrifice and get the B cells from spleen and fuse with the myeloma cells and make a hybridoma. And then only the hybridoma that secrete the antibody that you want is selected. And this process is very long and it costs a lot. Of course, this is actually got very famous and contributed a lot, and two scientists got a Nobel Prize in 1985. On the other hand, relatively new, as a relatively new technology, so someone tried to use phage to display the antibody on the surface of phage. And then after biochemically the process for binding, and it caused panning, and then only the highly bound antibody can be enriched and finally screened. This also will be for this technology. So two scientists got the Nobel Prize, George Smith and Gregory Winter in 2018. But still, a long time and big money, and selection, it's pretty much artificial. And the antibody is produced by first as a mouse sequence. And then that means we have to change the mouse sequence to human sequence to use as a medicine. Okay, so why it's difficult? Critical reason is that the B cells cannot be cultured at all. Only human B cell can be immortalized by EB virus infection, but its efficiency is very low. So, but if we can amplify the antibody gene from single B cells in a hybrid manner, we might be able to find the antibody gene and antibody protein. To do that, we have been tried this system for a long time and find eventually we can make it. We named it as EcoBody technology. So first, you get the B cells from immunized animals or human, and then enrich or selects the B cell that displays the antibody that can bind to the antigen. Now, this is the photo of the B cells and antigen coated Bs. So you can pick up a single B cell. And from the single B cells, we amplify the messenger RNA using RT reaction and several steps of PCR. And eventually, we make the gene, amplified gene, and then expressed using cell-free protein system. So then we check up the antibody binding using ELISA. The total process actually finishes in 1 or 2 days if you use a kind of robot machines. So, and after that, maybe the protein is antibody if we produce in E. coli or or sometimes we use mammalian cells. So, okay. And then also we don't, but just changing the PCR sequences, we can amplify from the human and a rabbit or mice. So it looks easy, but actually it was difficult. Of course, the PCR technology from single B cell is, of course, not easy, but we can finally find a good condition from the very small amount of messenger RNA copy number. And in addition, the expression of self-resist, of antibody, in this case, we express it the FAB. To make it efficient, we actually established 2 techniques. One is named zip body. The reason, light chain and heavy chain should be associate each other in natural form, but it will be done efficiently only in the mammalian cells. If we use an E. coli cell-free system, no chaperone or exists, and then association is not efficient. Therefore, so we add a Leucine zipper for light chain, heavy chain to help this association. And also the expression of the antibody, amount of antibody is also a problem issue. Because this is whole the sequence is animals, human sequence, and then normally it was bad. But we found out N-terminus small peptide, 4 amino acid SKIK sequence can increase the expression level in cell-free system very much. 40 or 6 times higher expression is available using just only 4 amino acid tag. And this tag is also effective even in vivo. This is the result of E. coli system. It also increased the protein expression. Okay, so combining this technique, the first expression level ability much increased by SKIK, and then okay, so without these tags, so this is the ELISA result. So this is the antigen, this is the RNA background. This level was very low But combining these two, you can see the high positive and negative control level is very increased. And then in addition to that, we are the SBC, is a kind of this SBC chaperone protein, then increase more much. Anyway, we can select the antibody binding activity amplified from single B cells. So using technology first, I tried to get a human, a rabbit antibody by ignite, started using using FACS screening, and we clone the antibody. In this case, we use magnetic beads, and then you can see this all the antibody is cloned. It show a very high signal to the target microbe. And after this protein is expressed in E. coli and solubilized and folded, and then the affinity is quite high. So, this is affinity. Sorry, it is using bio Brett sensorgram, and affinity is relatively high. It's pretty much level of the sub-nanomolar orders. Okay. So, and then we try to get human monogram dozen using test technology. So actually, it's not easy to get a human sample. So we got an EV virus infected human, immortalized human beetle from Riken in Japan. And then, of course, we don't know this, no information about this, the donor. And then maybe we thought that this, anyway, most of the people infected with influenza sometimes. So we tried to get a monoclonal antibody, human monoclonal antibody using influenza protein, vaccine protein. And first of all, we isolate the single B cells using the same magnetic beads technique. And only 0.1% B cells were collected. From the collected beads, we amplify the antibody genes and express, and then examine the binding activity using ELISA. You can see all the B cells antibody isolated from amplified from the single B cell shows some signal, good signal. That means I think we almost succeed to establish this technique, not only for rabbit but also human. And you know that the human monoclonal antibody should be very important because it can be used as a kind of the potential drug, and probably it may be useful for human disease or cancer or disorder. And me and Dr. Kato made a startup named I'm sorry, this is Japanese. So actually, and this is and also we got investing money from venture capital. Okay, so it works. I hope it works. Actually, so to demonstrate this technology, we made a video for you in Japanese first. But so only Japanese is available. I translate it to English by myself. Be patient for a while.
Let me introduce the body technology to obtain useful antibodies from rabbits and humans in a hydrophobic manner. First, a small amount of blood is collected from a human, mostly a patient or a rabbit, immunized with an antigen. The B cells in the sample are fluorescent labeled and sorted by a cell sorter and are delivered one by one into a well. Antibody DNA derived from single cell is reverse transcribed from each B cell in a 96-well plate and amplified by PCR. At the same time, two unique gene tags are introduced. The resulting modified DNA is used as a template for transcription and translation using a cell-free protein synthesis system to obtain the target antibody. The modified antibody then is not the whole antibody, but only the FAB fragment containing the antigen recognition site. The introduction of a leucine zipper tag at the C terminus of light chain and heavy chain allows the two domains of FAB to efficiently associate even in a cell-free protein synthesis system, resulting in the antibody with antigen binding activity. Introduction of the N-terminal SCIC tag ensures high yield of FAB fragment from any clone of antibodies. With this technology, FAB antibodies that can be used for various antibody evaluation, such as fluorescent staining. We can do it in only 2 days from the isolated B cells.
Okay. Okay. So now is now it is the homepage of iBody. Actually, I already leave as an CTO, and then this company still going on after getting more grant, more investment. And then fortunately, using this technology is a diagnostic company make a COVID-19 test kit. Okay, and also the company collaborated with medical schools and then found the human antibody from patient of SARS-CoV-2, and then they analyze, and these are our antibody work as a neutralized antibody. They might be a future medicine, and also they analyze the antibody, sorry, epitopes. Okay, and so our laboratory is still working on this technology, and one of the interests is actually, so how about the relationship of the intestinal microbiome and immunoglobulins? Recently, some paper published that about the animal might have the antibody against this intestinal microbe, but not so many. Therefore, we try to isolate the monoclonal antibody against these microbe using our technology. So this is one challenge, that we isolate the intestinal microbe from a rabbit, and also we collect the B cells from a rabbit, and then try to find the microbe-specific antibodies. And then after selecting it, we want to make it in a large volume in a low cost. Therefore, we try to use a blood batches excretion system, because Professor Tsumoto in Tokyo University already succeed the good expression and the purification. Therefore, we followed his protocol and make an expression vector and transform it into blood batches oceanosis, and then cultured, and then this is the Western blot, and the FAB is well expressed and the outside cells. So after that, we purified using histag purification. So, and eventually we purified using the size exclusion chromatography, and you can see this fab, only fab, and this is well expressed, very purified using separate system. Using microbe system, and measure the affinity using blitz the biolayer interference biosensor, and determine the KD value. Interestingly, this antibody binds most of the microbe, not only to also say, gram negative, gram positive. So there we are interested in the molecule that this molecular antibody binds. Okay, in addition, we introduce a kind of a challenge. Monoclonal antibody against GPCR. You know, GPCR is a very good target for many from many point of view. Of course, the structural analysis, and of course, this is a very good target for other medicine. But it is not easy. Still not easy because so it's hydrophobicity. It's difficult to purify, and immunogenic region is more, and it's hard to triggers immune response and lower expression. And so we try to express using the DNA immunization. So one idea is first make an expression vector and confirm the expression using mammalian cells. And using the same vector, we immunize DNA, vector immunize the rabbit and get B cells. So after that, so we isolate the single B cells that bind to the target. The target is actually use transfected mammalian cells expressing this GPCR and binds to the beads, magnetic beads using the tag system. Sorry. So we isolate the single B cells and try to express it, and then are actually some very relatively short, but we found some potential candidate of the antibody. But after that, we prove that after analysis, so it looks the bind this GPCR, but it doesn't. Oh, sorry, this doesn't work well. So I would like to now the Duffer is trying to do it again. I hope after he come back from Indonesia, he will continue this trial with a different challenge, with a different method. So, okay, so then I'm going to talk about the different technology. So this technology is called a single molecule display. So usually to this as a display technology, phage display is very famous. You know, phage is DNA. Oh, sorry, this is not good. So DNA phage is DNA is introduced to the E. coli and infected the phage, and then on the phage incorporate the DNA and the protein on the surface. So this is quite useful. And then 2018 to Nobel laureate got the Nobel Prize, but making this technology. But still, the library size is 10 to the power 9 or something, and depend on the E. coli system. If the expression of the protein is not good in E. coli, and also it transformed translocated the membrane. So this it might be cause some bias. But on the other hand, if we use cell-free protein synthesis, only the protein just synthesized and can be displayed on the linked, can be linked on the messenger RNA. Like one way is ribosome display. In the case of ribosome display, and the messenger RNA and protein is linked, physically linked via ribosome. The ribosome is somehow, and that means somehow only single messenger molecule can be linked to the protein synthesized. Therefore, so library size is so huge. Even in a 1 milliliter reaction, 10 to the power of 13 or 14, or much more library size is available. And also, now using a pure machine linker, and pure machine linker is chemically attached to messenger RNA, then synthesized, and then pure machine is incorporated in the protein. And then chemically link is established. And this link is very stable. So the library site is similar and very stable link. And actually, our laboratory use this technology to screen the antibody or peptide. Or now we're currently trying to screen the enzymes. So this is the kind of combination of cell-free systems. First, for example, we try to get an antibody using ribosome technology or ribosome display technology or EcoBody technology. And then, okay, this is a kind of our proposed kind of image of proposal. First, I would like to analyze the epitope very efficient way, because once you get an antibody, it takes a long time and it costs a lot to determine the epitope. The epitope is critical. Even the antibody is clean, but if the epitope is not good for example, biological function, it's meaningless. Therefore, we'd like to establish the high throughput epitope mapping technology. So we use the cell-free expression and the ribosome display and using NGS analysis bioinformatics to identify the epitope. So now we tried a kind of model system, and it works very well. Our target this day is anti-his HA tag antibody. Anti-HA is somehow the linear peptide sequence is the epitope. Okay, so now of course, in real, somehow the confirmation epitope exists, but as a first step, we tried this antibody, the HA tag antibody, only linear epitope. So okay, so first we make an NNK to the 10th power. It's a very huge DNA library made using random DNA sequencing and binds to the target antibody, and then 3 rounds was carried out. And after that, enriched DNA sequence is analyzed using NGS. So okay, so then interesting. Okay, so sorry, and then only as you can see, the very and highly okay, this sequence is enriched. Okay, this sequence is enriched very efficiently from the top, but you can see some. This is the HA tag. The only 3 amino acid is included. So, but it is very strange that this antibody can recognize only 3, because it should not. This antibody should recognize much more sequence. Otherwise, this sequence cannot be used anti-tag antibody. So we analyze this NGS data using a kind of original bioinformatics. Make it what we did is actually we pick up 3 amino acid combination and calculate the enrichment factor for 3 amino acid, right? And make a big a matrix, and then pick up assess the calculation index sum up, and you can see this is the sequence of the target hemoglobin, and you can hear the higher signal appeared. And if you look at this, so this is actually the HA tag sequence, and then you can see this is an okay, some amino acid is a high peak and some amino acid is a low. So we analyze we make a library amino acid representation for alanine scanning to see which sequence is important for antibody binding, and determine experimentally. Okay, this is the result of the expression level of the recombinant protein using cell-free system, and also this is the binding, Western binding signal, and this is the relative value. So higher relative value show that this is not recognized, but the lower signal value is recognized. And this pattern is pretty much matched with the calculation. So that results show that this method is quite useful, actually useful to analyze the binding site pretty much efficiently. We can probably, this strategy can probably used for the epitope mapping. Okay, so I'm going to talk, change to the new topic about the CDN display and enzyme, especially in this case, the enzyme target selection, screening. So here is a transglutaminase. Transglutaminase chemically bind lysine and glutamine and make a peptide bond. So this enzyme pretty much important. One is actually transglutaminase is now currently produced as a meat glue, and is used to make some sausage or something. And also, this enzyme is scientifically very important because this enzyme is expressed on the surface skin, and this reaction makes your skin much harder. So there are several transglutaminases exist, and we don't know very much about the sequence specificity of each transglutaminase. Okay, so what actually this research was done at Yasmina Damaznovich, associate professor in my lab, and she made the kind of DNA library and then transcribed and the messenger synthesized, and then introduced a cross-linking, the puromycin linker, anine, and then cross-linked and purified and used cell-free protein synthesis. And then in this case, a peptide is made. So in this, we can see this peptide and is linked with a messenger RNA, and it's transcribed and like this. And then you add a transglutaminase. Sorry, our location is not sorry. And then transglutaminase can make a peptide bridge between the library peptide and this is a chemical target of amino acid, insensitively lysine and different amino acids. And then this biotin is linked, therefore, so only the good substrate can be selected using magnetic beads, using this averaging biotin binding, and washing our way out that the library does not linked to the beads. So after several round selection, NGS analysis was carried out. And this is a summary of the results. This is a kind of the heat map. And then glutamine is fixed. And around this glutamine, what amino acid is preferred? Okay, so light color is preferred amino acid, and dark color is not preferred virus, and then only run an NGS sequence and the biofactor analysis, we can know which amino acid sequence is preferred or not preferred. It's pretty much efficient method to analyze the and also using this analyze the preference, and also using this analysis we can able to find out a good substrate. That means good substrate means a good substrate for this transglutaminase. This also this is a consensus sequence, and higher a good means somehow you can use it to detect the enzyme activity on the skin. Okay, and sometimes it's very important to find to distinguish the activity of EG1, EG2, 3, or something. And also, interesting thing is that using this data, preference data, we analyze whole the protein sequence and then rank using this matrix data. And interestingly is that, okay, so this is a higher rank that is already known proteins is detected as a higher ranked proteins, and also some new means somehow this is not regarded as this substrate, but using this analysis we found a candidate, neurofibrin is a new candidate of the TG1. Importantly, TG1 is known as a TG2, but not as a TG1. So we are very much interested in which is actually this TG1 is a target or not. And currently, we want to use this technology for the enzyme itself. In this case, enzyme is linked to the messenger RNA using the same pure-machine linker. And based on this enzymatic reaction, we'll be able to select the very high activity to the some designed the target. So now we are currently trying this process, this system. So name smart system. Probably we'll be able to get a very large, huge library. Theoretically, the most efficient because only single molecule is connected to the protein. Okay, so this is the last topic of our group. In the antibody selection, I talked about N-terminal SKIK that increase the protein expression level very easily, very greatly. You can see this, this is one example. And we examine so many cases, many tried many sequences. And so far, even some change of codon is not change the expression level. So we think that codon is independent. And the only translation is enhanced. And then it works as nascent peptide. So that means it affects only the protein on the cis mode. And interestingly, that this peptide can cancel the ribosome arrest. So this is a very new finding. So maybe some people don't know what the ribosome arrest. So this is a kind of very current topic about the ribosome protein. This is now very recently Nature Communication, in Nature Communication, some German group published the 3D structure of the ribosome arrest and the ribosome. So originally, this is found by Professor Ito, the University of Kyoto or Kyoto Industrial University. And their group found out the SEC protein is regulated by the SEC-M. But interestingly, that in the SEC-M sequence, and there is one somehow very short sequence that at this point ribosome is arrested and no more protein synthesized. In this situation, protein downstream SEC is expressed. But if not, that means if the protein synthesis is okay, this is normal case though. But if this arrest is cancelled because this is a signal peptide, and signal peptide go into the secretion machinery, this is pulled, and then protein and this cancel does not occur. In this case, so SEC ribosome sequence, so our ribosome binding sequence is anil covered by messenger RNA. Therefore, protein SEC is not translated. And anyway, in this case, so if you add the SEC-M sequence, SEC-M sequence and protein exception level is decreased. So we may, so SCIC, okay, can increase expression level, make it simple, okay. But the RS peptide decrease protein synthesis. If these two peptide, short peptide, put in the same protein at the same time, what happens? We try, and then interestingly, and of course, line A is AP is RS peptide. SEC-M AP is SEC-M AP sequence of that residue. Therefore, so SEC-M AP is a whole SEC-M sequence. But anyway, this protein is not well expressed in vitro. But if we put them as peptide sequence, the N-terminus, you can see some band. And of course, this is a normal impression, but if you add an AP sequence for the 4 or 5 in the top, no protein. But put SK sequence on the N-terminus, yes, and bound appear again. And interestingly, not only to the N-terminus, but also the internally put the SKIK in front of the AP, the band appears. You can see this. So, but if you put it in the 4, 9, and 10, and AP and the SK, if you put in the SKIK downstream of RS peptides, it doesn't work. No expression. Therefore, only the upstream SKIK can cancel the effect of RS peptide. And if the distance is shorter, this is long shorter, but if the shorter, the effect is more efficient. Okay. So what actually we have more data and might be published very soon. But this is a kind of the image that this is SEC-M sequence. And around this proline and the glutamine, this is already known that this this form, the translation arrest occurred. But somehow escape is on the same peptide. This might be pulled, pulled, and then this arrest will be canceled or replaced. So we actually found other similar peptide, and then these peptides can increase the protein expression very much. It depends on, of course, depends. Some protein cannot be very well expressed, and if you try this sequence, then you might be able to find good expression. Okay, this is the last slide. So So remember, so here is Sun and Kyril, he is contribute to our group, and also the first three guys from Indonesian student, and Dr. Semina, she contributed to the senior display, and Dr. Kato is contributed to the antibody selection, and also the SKIK translation enhancement research. Okay, thank you very much for your attention.
Okay, thank you very much for your interesting talk, Professor Nakano. It's quite new actually, I guess. So, well, Bapak Ibu, now we come to question and answer session. I hope maybe you can ask some question for Professor Nakano. So you can please raise your hand or you can just give any question in chat box. Any question from audience? Or maybe for enzyme groups? There's maybe some question about this new platform. Okay, maybe I will ask you some question. Maybe it's quite okay. There's someone who raised hand, Pak Faisal. So yeah, maybe you can open your mic. Okay.
Okay, thank you for the chance. Am I audible? It's Ninas. Am I audible? Can you hear my voice?
Yes.
Yeah.
Okay. Okay. Thank you for the chance to the video Nakano. Thank you for the interesting presentation and interesting research. My name is Faisal. This is a new research topic for me because my background research is about enzyme production by microbe, and never know about free living cell expression before. And what I want to ask is, mostly from your presentation about cell-free protein expression, have many benefits for medical research. Is that possible to have research about enzyme for bioenergy? Because that's about my background research. Thank you.
I'm sorry, I cannot hear you very well. So could you repeat again?
Okay. May I audible now?
Yeah, maybe better.
Okay. So my question is, from your presentation, cell-free protein expression have many benefits for protein medical research. Is that possible for us to have research about enzyme production for bioenergy?
Bioenergy?
Yes.
Yeah. Okay, so good question. So actually nowadays, so our cell-free system is the cost of cell-free system actually high. Therefore, so it's not practical to use it directly for energy production, but the system works well for just a selection, screening. For some, if you want some enzyme much more efficient, and then we'll be able to find out the good enzyme in using this system, and then it can be transformed to in vivo system. Maybe you need to use in vivo system for efficient production. And also, in the end, we just talk about the translation enhancing tab, and it demonstrates only the data about the cell-free system, but actually it works well in the cell system, in vivo system already. Therefore, so if you have problem with the expression of the protein in vivo E. coli, then the short peptide tag might work well to enhance the productivity of the enzyme in the cells. Might be used for energy production or something.
Thank you for the answer.
Is it okay for your question?
Thank you.
Welcome.
So maybe to continue the question, I would like to ask again. So this platform actually for this pre-screening or screening for further efficient production in vivo. I mean, at the end, we still need the production, protein production in vivo, or it's only for this kind of only for what's that, bioenergy enzyme, or for medical we can use directly for efficient production actually. Yes.
Yes. Actually, so we did not this research, but I introduced the one biofarmer, biotech, Sutora biofarmer established by Stanford University. The main one of the main technology is to make a pharmaceutical protein using cell-free system in an efficient way. So now the cost of cell-free system is greatly decreased, and then in future, if the cost is not too high, the new system might be compatible with a in vivo system.
Okay, thank you. Okay, maybe I would like to ask, actually this is quite new for me about this field, but maybe I will ask some question, some stupid question, sorry, Prof. Nakano, if my question
Don't worry.
too swallow. You said that for when you use this for epitope mapping with this I think it's using mRNA cDNA display, if I'm not mistaken. So you did like this epitope mapping by random library screening.
Yes.
It's actually why, I mean, it's such kind of libraries work actually, right? Library screening. It's any other way, for example, this directed approach to have this to near correct epitope. I mean, the target epitope. So we don't need to make this random library screening. So we can use like, I don't know, maybe from the structure-based information. So directed evolution maybe. So yeah.
I said, one but traditionally, for example, to make an epitope, there are several ways. One is the peptide library already, peptide array. One is to use peptide array, and one is use somehow protein recombination, and then the target protein is mutated, or more structured methods like NMR or deuterium, something like this. But all the method is it's time consuming and cost a lot. But if method works well, I mean, once a library is made, we can use it.
Okay.
You don't need to make a new one and analyze a different antibody and just selection and just NGS sequence. And the NGS sequence cost is becoming lower and lower. Therefore, once it's established, this is more efficient than previous ones. This is my not yet just started, but I think this our final object.
yeah, even though this from the first beginning, this very hard. I know this like very laborious work, but still we can like generate this from this library the information, so we don't need to repeat again. Yeah, so once library we get, so we can use for further work, of course.
Yeah.
Yeah, thank you. And then I wonder this platform cell-free protein synthesis, it's more but application in biomolecules. Is there, could we also apply this for screening to with the like molecule derived from natural products, for example? Maybe, yeah. Instead of beside antibody enzyme, could we also use or apply this for molecule derived from natural products?
I mean, you make a natural product using cell-free system. Like that?
Yeah, for biosynthesis, like if we have this biosynthetic gene cluster, we have this to express. Yeah.
Okay, so I'm my understanding is right on that, but actually one of the startups made by Jim Schwartz, now not Jim Schwartz, Dret is a Stanford professor, young professor of the Stanford University, as a specialist in synthetic biology. And his story is that all the protein enzyme is expressed in a cell system and combine them and then make new chemicals. And this process is, they say, much even faster than to make us from the viewpoint of synthetic biology. Synthetic biology, introduced many all the genes in the one cells and try to make it. But our optimization not easy. Okay, we don't know all the things is well expressed or not. But his strategy is just synthesize all the gene and cell system, combine these to make a new product. That is a kind of, as far as my understanding, I'm not sure, but this platform will help since the establish of the synthetic biology. So they think they make a company and already sell it. So that might be, he got a big money.
Okay, thank you. Okay, I think you start also your startup company, right? Since antibody Inc. And then you use this what you call it? Sorry, EcoBody technology, right? You use E. coli as a platform for the cell, right? And I mean, I think there's another platform not only use E. coli, right? Maybe also use yeast or etc. And then I mean, what's the advantages use this E. coli platform over the other cell platform in this case?
Yeah, actually, so expression level of protein using E. coli cell-free system is much higher than others. And that is a very important point. And also, we use a purified, we constitute the E. coli system. And then all the component necessary for translation is purified, and therefore, though no RNAs or protease is not included. That is very advantageous than others. Okay.
Have you ever compared with others, or I mean, it's this? I mean, before you did to others host also. I mean, for comparison, or it's based on the literature review or others colleagues that ever failed with that platform, or it's also depend on the what's the protein or enzyme that you use.
In the case of the antibody, this platform works very well. I'm sure, yes. But most of the, we also try several enzymes, and it works very well.
So actually, Yeah, it's similar to in vivo expression system also. Yeah, using E. coli is very, I mean, more friendly.
No,
to-
yeah, yes, that's
for
and
organ.
also in sometimes E. coli in the expression E. coli fail the active protein expression because of the inclusion body or no expression, but this sulfur system using sulfur system, I think the ratio of successful expression is much higher than in vivo system.
So this another advantages use this platform instead of the in vivo expression system, right? So what else? Maybe others. Okay, from we back to the room. Okay, to Miss Akhirta Atikana. Please, you can open your mic and then ask question.
Yes.
But mention where you're from. Okay, please.
Yeah. So I have question with you mentioned about the gut microbiota. So could you please elaborate more on that? Is that, I mean, if I'm a wrong interpretation, do you use this antibody to alter the microbiome, or you take some of the microbiome to produce the antibody? I'm kind of missed on that part.
So actually, this is a kind of a new trial, I think. So final goal is I want to isolate the human monoclonal antibody against the human microbiota. Therefore, it's not, it's almost difficult. But using technology, we believe that it's relatively easy. So that's why, as a trial, first we try to isolate the rabbit monoclonal antibody. But now we once tried, already tried to and succeed the human monoclonal antibody binding to intense microbe from EV virus transform cells. The kind of sequence is human. But now we'd like to make it from, especially for the milk, the mother, for example. It's my the next try. I really want to know what's the relation. There is no, yeah. To tell you, I'm not specialist of this field. I'm not the researcher of the microbiota. But when I read the microbiota, this microbiota affect human immune system, it might be true. But very few, antibodies, especially for the human, there's no report. So I really want to analyze or do some research about it. That's why we are doing right now.
Thank you.
Thank you very much.
Okay, thank you for your question and answer, Prof. Nakano. And then maybe other questions? I think, Professor, this is quite interesting and quite new from for us. So actually many questions, but like, yeah, hard to really deep understand. But it's very interesting then. So with this system, you already like have this if I'm not mistaken, so you already developed EcoBody for usually for antibody, right? And then and then smart technology for enzyme evolution. So many is like industrial enzymes. If not, did you know?
Yeah. Actually, now for the enzyme, we are dealing with the phospholipase D, for example, and transglutaminase. And transglutaminase, now we are currently express the microbiome transglutaminase. Microbiome is already, in the market.
Already in the market.
And I would like to use this technology to change some for example, specificity of transcriptaminase of using this technology, a kind of a protein engineering. We like to use it for protein engineering field.
Oh, it's very amazing. And then the third one is like translation enhancing peptide. It's quite new, yeah. It's the initial one, or it's already approved.
Yeah, actually very new, and yeah, actually we published in 2017, but nobody believes that, and because
Not yet,
but very new, but
yeah.
actually, so now we collaborating with a cryo-EM group in Japan, and why? Yeah, because on ribosome and ribosome LS peptide complex already analyzed using cryo-EM. So, but what happens if we add an SKIK? So we already start analyzing the complex, SKIK and RS peptide and the ribosome. So it's quite new, and I think it's a really exciting field.
It's actually this challenging, yeah. I mean, we need you need to further investigate the structural biology of this system, this ribosome system. And I'm wonder actually it's interesting. I think you develop your startup antibody in how long? I mean, from the lab into this.
Yes. Actually, so we made this company in 2018 February, but the technology itself is actually when we thought, the single cell technology is 20 centuries, almost before 20th century, we thought, and actually so published several papers, but it didn't actually go well very much because of the low expression levels. And very recently, 2016 or 17, we can increase the expression level very much, and therefore it becomes a real technology to make an antibody.
So this
took a long time from the idea to the reality.
But that's very interesting, Professor. I mean, from lab into the industry, that's actually what actually our ultimate goal, right? To be useful for others. And then, okay. I think we have one question from the chat box. This from Mbak Satyawati Triastuti. The question is why N-terminal is selected to produce antibody. Thank you.
Selected to produce N-terminal. Okay. The reason that N-terminal is not incorporated in the structure, it is free. So therefore, especially for short peptide, it will not affect the binding activity of the FAB. That's why we select N-terminus and and shorter one. If it's too long, might affect, but so the short peptide will not cover, for example, binding of the FAB.
Okay, thank you. Is enough, Mbak Satyawati? Okay, thank you for the question. And any question? So yeah, I guess if there's any question, thank you very much, Professor Nakano. It's very fruitful, new knowledge, and then very interesting talk, and then it gives us new insight into another approach and a platform, and then really nice to learn from you to make this from lab into this industry. Yeah, it's very nice from you. Okay. I think this talk from Professor Nakano that he worked on this cell-free protein synthesis, and he has like three platform, the technology in antibody for enzyme, and also the latest one about the transference enhancing peptide type. It's related to ribosome arrest, and it's quite new and still hoping to further work on it. And then I guess I hope this presentation will be very beneficial for you, give your new insight, and thank you for attention. Thank you, Prof. Nakano. And then I will give back the closing session offered to you, Mbak Suci, to the MC. Okay. Assalamualaikum warahmatullahi wabarakatuh.
Thank you.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mrs. Linda, for leading a very interactive discussion. And thank you, Professor Hideo Nakano, for a very interesting presentation. And as a token of our appreciation, we will give a certificate for Professor Hideo Nakano as a speaker. Thank you very much for sharing your valuable knowledge. Okay. Thank you,
Thank you very
Professor.
much.
Thank you once again. Ladies and gentlemen, we have finally come to the end part of this webinar. Before I close this session, I would like to ask Head of Research Center for Applied Microbiology, BRIN, to give us some short closing remarks. To Dr. Ahmad Fatoni, time is yours.
Okay, thank you everyone. Thank you, Prof. Nakano, for your insight full of new knowledge. But I hope that we can continue discuss on site to identify whether we can do some collaboration together with your team and our team and everyone here online. If you are also interested in collaborating with Prof. Nakano, so please contact one of our research writing so we can work together, not just between Research Center for Applied Microbiology, but we can expand to other research centers that are also related to the topic of protein synthesis for antibody, enzyme, and peptide enzyme. Because we know that we have research center for genetic engineering of Nakano. We will introduce a bit about BRIN later on after this meeting, after this webinar. I'm sure that many researchers from other research center who are working on this field are not attending this webinar. Later on, we can continue in our discussion to follow up from this meeting. Okay, thank you everyone for your attendance and also discussion. And I hope that we can gain new knowledge, everyone, about the protein research that has been done by Prof. Nakano and team from Nagoya University. Okay, I hope to see you again in the next webinar from Research Center for Applied Microbiology. So I close this session. Assalamualaikum warahmatullahi wabarakatuh. Thank you, Ms. Uci.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mr. Fatoni. Ladies and gentlemen, this is the end of our agenda for today. I'm Anu Lucy Arini. On behalf of the committee, I would like to say thank you very much to our speaker, moderator, and all participants. Once again, you can watch this recorded webinar on YouTube PR Microbiologi Terapan BRIN, and please follow our social media on Instagram at PRMT_BRIN and Facebook Pusat Riset Mikrobiologi Terapan. See you in the next event. Assalamualaikum warahmatullahi wabarakatuh. Thank you.
Thank you, Bapak Ibu semuanya. Thank you, everyone.